Hello and welcome to Unit 9, or excuse me, Unit 7, Day 9. We are talking about genetics still, and this final day of our genetics notes is about genetic engineering. And this is a major topic in history and in science right now. So this is really what that Vox Explained Designer DNA whole documentary was about, was genetic engineering. And genetic engineering is one of many types of biotechnology. And today's aim says, how can biotechnology increase our quality of life? So human beings always want to optimize science and get it to work for us. And we'll see with the case of biotechnology, it has been variable. And I will also share with you a little bit about the limits of biotechnology based upon some things that I had sent to me by some students in class. So stay tuned. But we'll start with the do now. Question number 1. Individual cells can be isolated from a mature plant and grown with special mixtures of growth hormones to produce a number of genetically identical plants. This process is known as cloning. So individual cells from a mature plant and grown with a special mixture of growth hormones to produce a number of genetically identical plants. We're talking about cloning. But we're going to go more into recombinant DNA technology today. So clones are genetically identical, and some cells can reproduce more quickly than others, and that is part of why we use them in our recombinant DNA technology. So to produce large tomatoes that are resistant to cracking and splitting, some seed companies use the pollen from one variety of tomato plant to fertilize a different variety of tomato plant. This process is an example of selective breeding. They are mixing pollen from one variety of tomato plant into another variety of tomato plant, but they're not going into the genes and splicing anything. Rather, they are actually fertilizing one plant with another. So that makes it a selective breeding. So the answer to number 1 and 2 on the do now is both A. All right, so then these are your notes under the first part. What is genetic engineering? So genetic engineering is the direct modification of an organism's genome. This is going beyond selective breeding. This is actually inside the DNA. And the genome is the list of specific traits or genes that are stored in an organism's DNA. Changing the genome enables engineers to genetically enhance desirable properties and to insert them into different organisms. Organisms created by genetic engineering are called genetically modified organisms, or GMOs for short. And the stuff in dark blue, I believe, is what you're filling in on your paper. So let's take a look at this technology in action. So we have here a plasmid, and that is just a word for bacterial DNA. Right? It is the genetic information of a bacteria. And this restriction enzyme comes in here and cuts the plasmid and makes a spot where there is a hole. Okay? So this hole has sticky ends that then this restriction enzyme is also going into foreign DNA with the desired traits. Okay, so humans are now manipulating this, removing that gene of interest. So this green part represents, let's say, resistance to pesticides, which is a real trait. that has been put in some wheat crops, right? So if the wheat plant itself is resistant to pesticides, they can use pesticides all over the crops and the wheat will still grow, and other invasive plants, weeds, will not grow, right? So they could take that resistance to pesticides and insert it in the plasmid. Well, now this is bacterial DNA. With the desired trait added. And what that is called, those two things being added together, is recombinant DNA. So it is a combination of the bacterial genome plus desired trait. So this is something human beings create when they want to make many copies of something, right? And you see we can do this through cloning. So human beings were really good at cloning bacteria because bacteria is easy. Bacteria does not have a sophisticated genome, or at least as sophisticated as humans. So we can go right into that plasmid and we can make many copies of this new trait. Bacteria also produces really quickly, and bacteria always makes genetically identical offspring, especially when they're being cloned like this, which means our desired trait isn't going to get mixed up in meiosis. So bacteria is a good host for this type of technology. This is why we use bacteria to create recombinant DNA, because it makes many copies of itself very quickly and the offspring are genetically identical. So it's not going to get mixed up through a process like meiosis. There's no crossing over happening when we're cloning bacteria. So now getting to addressing some of the silliness that I got sent by students in my 7th period especially. So could Spider-Man be real? Could we take a human and put spider genes inside of a human to get Spider-Man? No, we could not. But they do have web producing goats. So spider genes in goats enable the production of spider silk in goat milk. But again, that is a very limited specific gene to produce silk that is modifying the proteins being produced. So this is somewhat more limited than what some students sent me on TikTok. So we're going to watch these during class next week, but this is not possible. And why? The reason that this is not possible, well, it's because hamsters and deer and chipmunk and pigs have very different genes. And so what this means is, sure, human beings could create recombinant DNA, but the recombinant DNA between these different species is going to create unpredictable results. We do not have the sophisticated technology to go in and manipulate the genes of two species that are so different from each other, especially because not everything is going to match up perfectly. A really good example of this is how a mule is produced. So we have here a horse and a donkey, which are very similar species. If you've ever seen a horse and a donkey, they look a lot alike. But, and again, these are very similar species, much more similar than, for example, a hamster and a deer or a chipmunk and a pig. They have a different chromosome number. Okay, so 64 versus 62. That seems really similar, right? But even in the case of natural production of offspring, so these two organisms can mate, they can produce an offspring, their offspring will produce what's called a mule. And what that means is the mule is not an actual species because it cannot have offspring. So all mules are sterile, which means yes, they did produce an animal, but it can't have any more babies of its own. And again, this is a situation where just if a female horse and a male donkey had sex and the female horse was to get pregnant, they could actually produce this baby, this mule. A male foal as well would be sterile. But it's saying in this report that, oh my gosh, maybe we did find one that was not sterile. But forgive me, I just got, in short, don't believe everything you see on TikTok. And in this case, this was a trick as well. You're only going to produce a female mule that is not going to be able to have any offspring. And what happens here is, for example, let's look at human chromosomes versus mouse chromosomes. And you can see that all of these are bound up DNA, right? So there are many genes here included. And let's say the genes for mouse eye color might be on this chromosome versus the genes for human eye color are on this chromosome. Humans do not have sophisticated chromosomal mapping. We do not have the genome mapped out for every species and understand exactly what each gene does well enough to actually produce, like that video suggested you might, make a striped pig by using a chipmunk pattern. No, in fact, as you can see, I found this just on Google by a quick Google search. This is a wild boar piglet. So we're going to watch this TikTok in class, but the chipmunk and pig hybrid is actually just a wild boar piglet that is a trick talk. Don't get scammed. No good. But genetic engineering is something that human beings have actually been doing for a long time. We use CRISPR today, but that technology is in its infancy. And maybe someday CRISPR will be able to produce the genetically enhanced hybrids that people are imagining on TikTok. But for right now, what we do have are genetically modified organisms that mostly make up fruits and vegetables that we eat, although they have genetically modified some fish as well that they sell as pets. So if you've ever seen these really colorful fish at a pet store, they are genetically modified to be those bright highlighter colors to sell to people. And that is kind of messed up, I think, but I suppose they are fish. If anyone's seen Seaspiracy, we should be caring about fish feelings, I suppose, but just an interesting side note, not really useful for food. But in 1973, humans created the first genetically modified bacteria. And then by 1974, humans created genetically modified mice. And it's actually those genetically modified mice that are used in most experiments that involve mouse subjects. So they have put monoclonal antibodies from humans into mice. And that is really where they test a lot of vaccines, for example, medications on these genetically modified mice that include monoclonal human antibodies that the mice grow. They have done things like grow a human ear on the back of a mouse. The hope is that one day we could grow replacement organs by using organisms like mice, but we're still far away from that technology. Again, we have access to this technology, but we are still developing it, which is why it's really fun to learn about it in science. So, the first commercial development of GMOs was insulin producing bacteria. So, this is where we get insulin for medication. And this was only in 1982. So, before that, people that had diabetes had to get insulin that was harvested from humans, which was much more difficult to synthesize and produce. But now we use bacteria to make that insulin. And you would think it would make insulin cheaper, but sadly, it has not. In 1994, we began to sell genetically modified foods, which are usually fruits and vegetables, such as corn, soy, tomatoes. In other countries, there is more restriction. I might have just spelled tomatoes wrong. In some other countries, they have more restrictions against selling genetically modified foods. But in the United States, we really don't, and they don't even have to be labeled. So even if you've been trying to avoid genetically modified foods, you have definitely eaten some in your life. So let's take a look at some examples of genetically modified salmon versus farm salmon. So we can see the genetically modified salmon measures 24 inches in length versus the farm salmon is only 13 inches. So this one is a lot bigger. The weight of the farm salmon is only 2.8 pounds, whereas the weight of the GM salmon, wow, it's 3 times as much, or excuse me, a little over twice as much. Wow. This fast-growing salmon combines genes from 2 other fish, which cause this salmon to continually produce growth hormones. This is, a boon for the salmon industry. This is making people some money. We also have less smelly cows that we have produced through genetic modification, which modifies bacteria responsible for methane production, aka cow farts. And this results in a 25% less flatulent cow, and that would actually be good for the environment. I'm recording this on Earth Day, and cow farts are responsible for a lot of methane, which is a greenhouse gas. So go ahead and have less smelly cows. Although, again, when we think about these things, how do those genetic modifications affect the humans that might be eating from these species? We really don't know. But again, if you think that you've avoided this, you probably have not. A lot of food that is sold in the United States is genetically modified, most of it, probably, in fact, and we do not have to label it. So, engineering plants. This is where you're going to hypothesize a little bit, think about how this technology can be used. Like I said, it is really being developed right now. So this is going to change the course of our lifetimes. So we see there, these are branded seeds. So again, there are a lot of people making a lot of money off this proprietary technology. How might genetic engineering modify plants to solve everyday problems? Consider world hunger, weather problems, insecticide pollution, climate change. Other reasons to genetically modify crops. So we modify crops to be insect resistant, so they can't be harmed by bugs, herbicide resistant, so they can kill all the weeds that are growing near the vegetables and still keep growing the vegetables. Drought and freeze resistant, meaning they can grow in any climate. Disease resistant, so they're not harmed by diseases. Higher yield, faster growth, improved nutrition, longer shelf life. All of these things are to improve the human experience without really very much regard for the organisms that we are modifying. But this is all to improve human life, which is, a good thing. But I've been hinting at it already. What are some concerns about GMOs? So, what are some concerns regarding genetically modified foods and animals? There could be a risk to human health if they are unsafe to eat. Again, we've already been eating GMOs our entire lives, but is that safe? We really don't know. In Europe, they have a lot more restrictions against eating genetically modified organisms, but in the United States, we've mostly been doing it without exception. And we're still alive, but that's definitely a question mark. Harm to the environment and wildlife. So we'll talk about some scenarios, but in general, what can happen is these genetically modified crops, they are disease resistant, they're herbicide resistant, they overtake the original species. And these are crops that are sold for money. So if it is found on your farm that you have this genetically modified strain that can be detected, it's unique from the natural version of, let's say, wheat, farmers can be charged by the company that sells those seeds saying, hey, you're growing our proprietary copywritten seed on your property. But as we talked about, wind just blows. So those fields may have been cross-pollinated by blowing wind, bringing the genetically modified pollen their way. And by no fault of the farmer is this now a copywritten product being grown on their farm. It's a bad time. Bad time. So again, for the most part, we're going to continue to explore this as a modern issue. Just like we watched that documentary, we may read passages about GMOs, so you want to put your critical thinking caps on. How do these things actually affect our lives? How does this affect human life? So increased pesticide use and herbicide use, because these genetically modified crops are being grown to be resistant to pesticides and herbicides. That means they are being covered in pesticides and herbicides because it's not going to harm them. It's just going to harm whatever they're not making money off of. And then that means that the herbicides and pesticides are making their way to us, to our food. And we will talk about bioaccumulation when we get to ecology, but this is a bad time. And again, we'll talk more about this in ecology. So seed and pollen drift, again, this is a big problem, and people wind up being charged money for that seed and pollen drift, meaning these genetically modified crops are growing places where they're not supposed to be growing. The farmer's health from increased pesticide and herbicide use, the creation of herbicide-resistant superweeds. So now these GMO plants are mixing in different ways because they can cross-pollinate, because nature is going to nature, pollen is going to go where the wind blows. And what about genetic engineering in humans? This is the big question, what we watched that Vox Explained Designer DNA about. Nearly 50 countries around the world, including Australia, Japan, and all of the countries in the European Union, have enacted significant restrictions or full bans on the production and sale of genetically modified organism food products. And 64 countries now have GMO labeling requirements, but not the US, because it's making some rich people a lot of money. Scary. Okay, so we're almost done here. Bacteria producing human insulin. So this is a great use of GMO technology. The accompanying diagram illustrates some steps of a procedure in one area of biotechnology. So we are taking the gene for human insulin, cutting the bacterial DNA, using restriction enzymes to insert that bacterial DNA back into, to insert the human insulin gene into that bacterial DNA, then insert that gene into the bacterial cell where it will divide, and the letter X is going to represent the bacterial cells that are able to synthesize human insulin. C, and this is, of course, an example of recombinant DNA. And cell division, these are all going to be genetically identical offspring that can synthesize insulin. Thank you, recombinant DNA. And this is how we get insulin to treat people with diabetes. So plasmid, this is the circular DNA structure used by bacteria. So it's not the double helix, but it is still DNA. A restriction enzyme is an enzyme that cuts DNA when specific base sequences are present. So it is specific. The recombinant DNA is the product. This is the DNA in which a section has been removed and replaced or recombined with a new sequence. So our example is here, where the bacterial DNA now has the genes to synthesize insulin, to produce insulin, and DNA ligase, which is an enzyme that combines DNA bases. So we talked about DNA polymerase that is involved in DNA replication, and now we're talking about DNA ligase that combines DNA bases. So, part C, the conclusions, describe the role of restriction enzymes in the process of transformation. So, restriction enzymes are used to cut the DNA of both the organism with the desired gene and the plasmid. This allows the fusion of the nitrogen base pairs in the two DNA segments. And again, why do we use bacteria to do this? It's because bacteria can produce many copies very quickly, and they're all going to be genetically identical. Last question. Whew. Describe how bacteria can be made to produce human insulin. First, a restriction enzyme cuts both the bacterial plasmid and the human insulin gene. Then, an enzyme called ligase joins the nitrogen bases of the cut plasmid and human insulin gene together. Thanks DNA ligase putting together that recombinant DNA. This creates the recombinant plasmid. Then this recombinant plasmid can be inserted into a bacterial cell. When the bacterial cell reproduces, it creates more cells that now have the recombinant plasmid and can produce the protein insulin. Thanks, DNA ligase. Thanks, restriction enzymes. Thanks, biotechnology. So this was the last day of genetics, where we talked all about genetic engineering. And our aim was, how can biotechnology increase our quality of life? And we learned about insulin, which is a good thing, but we also learned about GMOs, which are an unsure thing, but are something that are present all over our lives, whether we knew about them or not. So I hope you guys have a super fantastic day. Good luck on your Unit 7 genetics exam. I'll see you guys all on Zoom. Bye. Bye, guys.